Hi again, everyone. Welcome to Chapter 17. This is Part A, Gene to Protein. Put another way, transcription and translation. So we have the mechanisms of transcription and translation. Transcribing nucleic acids into another nucleic acid and translating that language of nucleic acid into a language of amino acid sequence, which leads to generally a functional molecule of some kind, what we call proteins or peptides or polypeptides. So as I record this lecture, a big deal has occurred in that there seems to be a potential therapeutic breakthrough for the coronavirus outbreak that we're experiencing. And they're using what is known as an mRNA vaccine. Well, this chapter is perfect for understanding what's going on with an mRNA vaccine. What they're doing is they're taking mRNAs that they've identified from the COVID-19 virus, also known as SARS-CoV-2. They've isolated some of the mRNAs in that virus, and they package it into basically an artificial membrane, if you will. It's usually a lipophilic type molecule that creates an envelope around the mRNA. Now, they only give you some of the viral mRNA. They don't give you the genes for replication. They don't give you some other things that would make this an actual virus. They're giving you some of the mRNAs that they think are less variable in this COVID virus. They package it in a membrane that will allow it to penetrate cells. And so this synthetic mRNA penetrates the cells, and the ribosomes in the cytosol of the cells will immediately begin translating this mRNA into functional peptides, which happen to be the viral peptides. Viral promoters, and we're not going to talk about promoters all that much. Promoters are sequences in the DNA and RNA that cause higher levels of expression, and viral promoters are the strongest we know in nature. Very strong. So once we get this mRNA inside the cell, the ribosomes almost can't help but translate this stuff. So they'll translate it into the viral proteins, but not an intact virus. And so the cells will release these viral proteins with a little bit of help from some of the sequences here, and your body will start generating antibodies, and not just one antibody. Like, for instance, you could give somebody what they call a monoclonal antibody, maybe just this guy right here, one clone of an antibody. But in the case of doing it this way, your body generates legions and legions of antibodies to different structures on the different viral proteins. Incredibly powerful, minimal opportunity for side effects. This could be the future. They were trying to do this with influenza. The company that first announced their results was trying to develop an influenza vaccine this way. They switched over to COVID-19 when it became evident that it was going to be a very serious problem. But now we have two companies that have released results that say 90+% efficacy, at least in the trials that they've done. That is tremendously exciting. And for not only COVID-19, but also other diseases that are viral in nature that have eluded traditional vaccines. So understanding transcription and translation becomes uber important, because I think this is the future, guys. I think this is the future. So here's just a little, I'm not going to belabor this too much longer. This is one of the most important things that happened, and it happened really quickly when SARS broke out, was that Chinese scientists and other folks shared the genetic sequence, and we sequenced the RNA in the COVID-19 virus at blistering speed, and we found these are called open reading frames, ORFs, okay, that will translate into different things, different ORFs, and then different little ORFs out here. ORFs mean that there's potentially a protein that's created. So potentially an mRNA that's created here or here or here. And so there's a bunch of them. And so they had lots of potential targets for encoding for viral proteins. So again, understanding the nature of transcription and translation becomes really, really important. And here's just a COVID-19 virus picture. You not only have the RNA in there, about 30,000 nucleotides, but you have the proteins, and each one of these proteins represents a potential antibody target. Maybe the phosphoproteins that cover the RNA and protect it inside the virus, or the spike proteins that are so visually noticeable, or these other proteins. What the companies were doing is trying to find the ones that were the least variable, or the ones that were the most immunogenic. That is to say, they elicited the strongest immune response, and they choose those for the mRNA virus, or for the mRNA vaccine. Now that you've seen where this can go in terms of application in our lives, why don't we first start by taking it from the top, making sure that we remember the central dogma, that is to say, DNA to RNA to protein. Proteins are the doers, right? DNA is the storage, and RNA is the bridge between doing in the cells and the information being stored in the cells. The link between what we call genotype, that is to say the genetic information, and phenotype, which is the manifestation of that in individuals and in populations. Phenotype are the traits, and proteins drive phenotype. Proteins drive phenotype. For instance, this young man's brown eyes are brown because of the presence of melanin in his iris. Blue eyes are generally absent of melanin. So in his genome, the melanin gene has promoters and enhancers in the gene that say express the melanin protein in the iris of the eyes. That's one example of millions of examples. And we're going to work on the basic system of how we get from here to here. Subsequent chapters talk about which ones are going to be turned on and which ones aren't. We're not going to get there this semester. Nevertheless, a big deal is understanding this if we want to understand the basis of genetic variation, differences in individuals, and or treat diseases. We call this process of turning a given sequence of DNA into a protein that manifests in a trait gene expression. How much protein from that original DNA sequence is created through the steps of transcription and translation. There are regulatory steps at each and every one of these levels. We're just going to work on the basic process in this chapter. How we got here, understanding the connection between DNA and transcription and translation, came about through studying metabolic defects, particularly in diseases like hemophilia. There are different kinds of hemophilia, and the different kinds of hemophilia translate into a defect in one of the proteins in this cascade leading to blood clotting. Many genes, the phenotypes are manifest because the protein that they encode is an enzyme that catalyzes a specific chemical reaction. Other phenotypes are going to be occurring because of the shape or the distribution of a protein, like for instance the melanin gene that I talked about in the previous slide. But the studies from these metabolic defects, we traced back to the inability to synthesize or the inability of function, loss of function of a given enzyme. We don't have blood clotting because one of these steps was interfered with. So linking genes to enzymes requires an understanding that transcription and translation is fundamentally dynamic. In addition to being a pathway to getting to protein, it's also a pathway that can be controlled at various levels, at the DNA level, at the transcriptional level, and at the translational level. It's a series of steps, metabolic pathways. Cells synthesize proteins and degrade proteins and other molecules that might be interacted with that protein in a series of steps. It's one great big collection of interactions, and that's the hard part to see through. So what we're trying to do is dial it back to a very, very specific pathway to help you get a start in a greater understanding that will frankly be required of you if you move into healthcare paradigms. We don't just see this in hemophilia. We actually see this not only in humans, but also in other organisms. For instance, we have a metabolic pathway that takes a precursor and creates the amino acid arginine. Certain organisms can synthesize arginine on their own. I think humans can as well. But it requires several steps. You have to first take the precursor and turn it into ornithine, and then convert it into citrulline, and then eventually convert it into arginine. And what we found were that there were different mutants in the model systems that we were studying that couldn't create arginine, but yet you could see what was present and what wasn't. So like for instance, didn't make arginine and all we saw was precursor. Didn't make arginine, but we saw precursor and ornithine, and so forth and so forth. And that allowed us to tease out the metabolic pathway that starts with gene A converting precursor to ornithine, gene B converting ornithine to citrulline, and gene C citrulline to arginine. So the missing arginine here was the start of the phenotype. They all had the same phenotype. But when we dug a little deeper into the cells and looked at the molecules in the cells, we saw a different array of molecules present in those cells. So RNA becomes the bridge, shall we say, between the genetic information in the DNA and the physical manifestation of that genetic information in terms of proteins. That process of creating RNA we call transcription, and it's under the quote-unquote direction of the DNA. That is to say, the nucleotide sequence in the RNA is a facsimile of the nucleotide sequence from the DNA. When we're making proteins, we're specifically using messenger RNA as opposed to other forms of RNA, which include ribosomal and transfer RNA. In fact, we think RNA actually came first. If we look at the evolution of life, because of the autocatalytic nature of RNA, we think that it actually preceded DNA. But in any case, once that RNA, and in this case messenger RNA, is created, with a little m there, messenger RNA, that then is turned into a amino acid sequence that constitutes the primary structure of a protein. And that occurs in what's called a ribosome, which is itself a protein complex. Ribosomes and the genes that encode ribosome, from ribosomal RNA and messenger RNA, are some of the most highly conserved sequences in all of life. In eukaryotic cells, messenger RNA has a lifespan of its own, and these lifespans can actually vary, and we can see changes to the RNA made after it has been transcribed from DNA. So when we transcribe the precursor of the messenger RNA, we call that a primary transcript. That is to say, there are signals in the DNA, and proteins will come in and turn and create RNA using DNA as a template. But even then, once we've created this, we call this the primary transcript, because we're going to actually process this transcript. We're going to remove segments. We call these segments introns, non-coding segments found inside a gene. And we're also going to do some other things to this RNA which affect its half-life once it's been created. In a bacterial cell, the route from DNA to mRNA to protein is much, much more direct. First of all, you're not dealing with a nucleus, or you're not dealing with different organelles. You're really only dealing with a functional inclusion called a ribosome. And in fact, You can be transcribing the DNA into mRNA and simultaneously translating that mRNA, even though it hasn't been completely transcribed. So you can see the mRNA is growing off of a segment of DNA and ribosomes literally on that RNA as it's being transcribed off of the DNA. In the case of eukaryotics, though, you have a primary transcript, a pre-mRNA, if you will, created. That mRNA gets processed. Some of the material will be clipped out. Okay, we have a clipping and an editing situation. But we also have modifications made to the top and bottom, the 5' and 3' end of that mRNA, to prevent it from being degraded too fast once it moves out of the nucleus and either into the endoplasmic reticulum or into the cytosol. The mRNA in bacteria and prokaryotes is not processed like in eukaryotes. It's a direct line where you transcribe and translate virtually simultaneously. But in the case of eukaryotes, we're going to make that primary transcript. We're going to edit out the introns. Okay, we're going to splice the introns out. We're going to add a cap, a special methyl cap at the top, and a poly-A tail. This is a tail of just almost exclusively RNA nucleotides. I had a molecular biology professor who talked about in cells there are enzymes that just seem to non-specifically chop up mRNAs, and he called them nibbleases. They tended to nibble at the top and bottoms of mRNAs. And so the cap and the poly-A tail are there to kind of stave off the nibbleases, if you will, and keep the mRNA active for longer once it's been created. Once that RNA processing has been completed, the mRNA is exported out of the nucleus. In eukaryotic cells, we make that primary transcript, we edit out those introns, cap and tail, export that mRNA out to where the ribosomes are located, either they're on the rough ER or in the cytosol for translation. Both of these processes, transcription and translation, have a set of definitive steps that we're going to drill down into later on in this chapter. The major questions was, how do we translate the DNA/RNA into amino acids? How are those instructions conveyed? Especially when you have 20 known amino acids, but only 4 nucleotide bases. Clearly, it's not one base per amino acid. The math doesn't work out. So how many nucleotides could it be? Is it 2? Is it 3? Is it 4? Well, I'll give you a little hint. They finally figured out that it was 3. And that some amino acids are actually special. Like, for instance, the amino acid sequence for methionine is actually also a start. We have stop codons as well. So it was one of the real great discoveries for unraveling the genetic code into how we sequence amino acids based on the nucleotide sequence in the mRNA. Following some very careful and powerful studies by a couple of researchers at NIH that I'll talk about in a few minutes, we have determined that it's a 3 nucleotide code. We call it a triplet. And that phrase that we use for that triplet code is known as a codon. So the words, if you will, are in 3 letters in the mRNA. And this is one of the charts that you'll see. You can see rectangular charts, and we also have these radial charts. What you do is you work from the center out, so going 5' to 3'. If the first letter is a G, and the second letter is a U, and the third letter is any of these 4, it's going to be a valine amino acid that's encoded. So we can then create a primary sequence of amino acids based on the linear sequence of nucleotides using 3 letters. Like, for instance, valine could be GUC or GUU or GUA or GUG. All 4 in the 3rd spot can work for valine. That 3rd spot, by the way, they call a wobble because it's a little bit less important than the first 2 letters. You can see the big first letter, the relatively big second letters, but the fact that you can see more than one in these other ones is to the fact that that 3rd one, because of the atomic shape and so forth, allows for a little bit of wobble in there, and different letters can create the same amino acid. Let's break this out a little bit. Let's say we have a gene in the DNA, and we have 2 strands. Now, in eukaryotes, it's very unusual to have one side be a coding strand and another side being a coding strand. In DNA, generally there is one side. It can flip-flop depending on which gene you're talking about, but there's almost always one template strand of the 2 in DNA. And so for our purposes here, we're going to say that the top strand here is the template strand in question. And so we're going to transcribe that into the complementary base sequence. And then we're going to read that complementary base sequence 5 to 3 and create amino acids based on the 3 letters. UGG encodes for tryptophan. UUU encodes for phenylalanine, and so forth and so forth. So transcription is creating the complementary mRNA to the template DNA strand. It's going to be one of the 2. It's not both. In bacteria and viruses, it's a little bit of a different story, but here in eukaryotes, it's almost always just one strand, translating it into primary sequence for proteins. I've already said this in the previous slide, but the thing I would like you to understand is that we're going to synthesize 5 to 3 when we're making this RNA. We're going to synthesize 5 to 3. So we're going to read 3 to 5 on the DNA. And we're going to synthesize 5 to 3 on the RNA. And when we translate, we are going to read and insert amino acids reading 5 to 3 as well. That directionality is very, very important when it comes to transcription and translation. We always synthesize 5 to 3, and we read 5 to 3 synthesizing top to bottom on the amino acids. And so here's just a picture of a codon, a 3-nucleotide sequence. And again, we've already talked about this, just giving you some additional pictures for that same story. And a really, really interesting story about how this code got cracked. These 2 guys right here were the guys at NIH who figured it out. And it turns out they were outsiders to this. Everybody thought that it was going to be Francis Crick or somebody from, the rock stars in the world that had uncovered the structure of DNA, but it was 2 guys. In fact, when they presented their paper about deciphering the code from their initial experiments, there were only like 6 or 7 people in the audience. It was lucky that one person in the audience knew Francis Crick and said, hey, these guys are onto something. And Crick actually arranged for them to give their presentation again, only this time to a room of hundreds and hundreds of people. And they ended up winning the Nobel Prize not too long after that. In any case, it was a very, very cool experiment. They would just incubate artificially created mRNA, like for instance poly-U. This is nothing but U across the whole thing. And when they did that, when they added it to an extract containing bacterial ribosomes, they got phenylalanine chains. Or when they did CCCC, they got proline chains. And that was the first step into uncovering all 64 triplets. 61 of the 64 encode for an amino acid, and the other 3 are what are called stop signals. That is to say, you're going to be translating until you hit this codon that says stop translating. And when we get to translation, we'll talk more about that. It's redundant. That is to say, more than one codon can encode a particular amino acid. And again, it's that wobble in that 3rd position that's mostly responsible for that. But some people have hypothesized that this redundancy allows for mutations to occur that then don't just shut the whole protein down. So by having a mutation in one amino acid, you may still be able to encode phenylalanine, even if it's not perfectly UUU, so that it will help stave off the influence of mutations, except in even more rare circumstances. But the one issue is that what happens if you delete or add a nucleotide accidentally? Since we are in a triplet, we have 3 and 3 and 3, but if we were to delete or add it, that would cause what we call a frame shift. That is to say, you're no longer in the right frame. There are 3 possible frames if you think about it. Do we start on this nucleotide? Or do we start on this nucleotide? Or do we start on this nucleotide? So all 3, especially if you have varying sequences after that, could be very problematic. And when we talk about mutations, we'll talk about reading frame a little bit more. So I'm going to stop for now. So this is the end of part A, and I will pick up on the details of RNA transcription, the regulatory elements in the DNA, the proteins, and so forth in part B. So thank you for your time, and I will see you again in part B. Talk to you soon. So in transcription, we have to start the transcription process from DNA to RNA in the proper place. Because if we just mentioned in the previous slide that reading frame is really, really important, how do we determine what the first frame is? And so looking at a breakout of the codons, and these are the first letters over here. So if you choose A for your first letter, and then you choose U for your second letter, and then you choose G, so you go U, and then you look for the G on the third letter, we find this really interesting codon right here that's flagged, AUG. AUG encodes for methionine. It's the only codon that encodes for methionine, and it also is the start codon. It is the very first one and gives us a potential reading frame. You might encounter, and we saw this in the virus earlier, you saw those ORFs, those ORFs, open reading frames. That's where somebody has scanned the RNA and found an AUG, which means that it's a potential start for a gene. We may not know what that gene does, or even if it encodes a gene, but we certainly can see the beginning of a gene and a potential reading frame. No other codon in all of these 63 other possibilities do we have a start. So here's a way not only to flag the beginning, but also to put it in the proper reading frame. And here's the fun setup. This is a sequence of mRNA for a gene that I studied as a postdoctoral fellow at Indiana University School of Medicine when I was over at IU. If we scan through this, you might find, and if you scan, you scan, you scan, oh, there it is, an AUG right there. So now we have the reading frame. So here's the first codon, AGA is the second codon, ACG is the third, GCG is the fourth, UCC, and so forth and so forth. There is our start codon. And you can see in this mRNA that we have that there's a lot of nucleotides upstream that are not going to be encoded in that gene. Part of the processing, if you will, part of the way that mRNA works is that we have to set the proper reading frame for translation. And so we have our AUG right here. You remember when we talked about DNA replication, we had DNA polymerase 1 and DNA polymerase 3. Well, for RNA synthesis, we have RNA polymerase. The interesting thing that sets RNA polymerase aside from DNA polymerases is that, first of all, it's making RNA. That's fine. It still reads 3 to 5, synthesizes 5 to 3. But RNA polymerase has its own helicase activity. Remember that we had to have helicase in order to melt the DNA strands, to pry those strands apart. So RNA polymerase, in the sort of the same vein that DNA polymerase 1 had exonuclease activity, in addition to polymerase activity, RNA polymerase has helicase activity in addition to its polymerase activity. It literally pries the DNA apart, it melts the DNA, synthesizes the RNA, and as the transcript is formed, it will begin to, the enzyme complex can actually remove that mRNA transcript, and it allows the DNA to re-anneal once it has passed through the enzyme. It's using the DNA. One strand is going to be the template strand. We're going to synthesize that RNA. One of the major differences in terms of RNA versus DNA, in addition to being single-stranded, is that RNA does not contain the thiamine base. It uses uracil as opposed to thiamine. So ACGU as opposed to A, C, G, and T in DNA. So RNA is single-stranded, and it has uracil base, pyrimidine, instead of thymidine. So in addition to that AUG codon, there are other sequences that help us properly transcribe only genes from our DNA. These sequences are often called promoters. That is to say, they're special nucleotide sequences that promote the association of RNA polymerase with the DNA and facilitating transcription of that DNA. On the other end, down at the downstream end, we have sequences that will promote termination, that will facilitate, I should say, termination of transcription. So we're not just randomly transcribing DNA any old place. There are very, very specific places where that RNA polymerase is going to bind, transcribe, and release. Put together, the promoter, the coding sequence, and the terminator we call a transcription unit, and this is functionally a gene. So a gene contains a promoter, generally contains a coding sequence of some kind, and most often, not always, but most often, has a terminator sequence at the downstream end. So we have the components for regulated transcription of genes at the appropriate time in the appropriate amount. So let's come back to our syntaxin 4 gene again, only this time we're going to be looking at DNA. Notice that we have T's in here rather than U's. Okay. Well, here's our ATG, which is the DNA equivalent of AUG in the RNA. So there's our start codon right there. That's the beginning of our coding sequence. But look at this sequence right here. TATA. That is known as a TATA box, this 4 nucleotide sequence. By the way, you can scan through here, and I doubt you will find another TATA box in this whole area. I doubt you will. There may be one out there, but doubt it. This is a major, major piece of the promoter that sets us up for transcribing the RNA that we're going to create syntaxin 4 protein, which is really, really important for insulin secretion, by the way. So we have that promoter, and that promoter is where the polymerase will bind and initiate transcription. And then we get down to the end of the terminator. Okay, so we're going to start transcribing, and here's our template. We're reading 3 to 5, synthesizing 5 to 3. We work our way down, elongating. As we elongate, that transcript is removed off of the DNA, and the DNA is allowed to re-anneal with itself. And this thing slides along and creates our primary transcript. When we hit that terminator, the RNA polymerase will stop transcribing. It will release the DNA, and the RNA will be released as well.